All right. Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us the 55th seminar. Oh my God, already 55th seminar. And I call it our Nature Microbiology Day. Last week, I visited Stanford University, University of Illinois, Urbana-Champaign, and University of Wisconsin-Madison to give seminars. I will continue my seminar trips right after this SynBYSS seminar today for roughly 6 weeks, with occasional coming back to my home until early December. So I will chair this seminar series in another city, probably mostly in Asian countries at probably midnight to 2 a.m. in my time there. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Susan Jones. Of course, she does not need any introduction because we all know her. She is the chief editor of Nature Microbiology, and of course, one of the top journals we all wanted to publish there. She is one of the 4 Nature journal editors who participate in this seminar series as pioneer speakers. Therefore, Nature journals have the largest number of people supporting this SynBYSS seminar series. So I and we tremendously thank her and Nature journals a lot for the time shared with us. So Susan, the virtual podium is all yours, and thank you so much again. And I will share for her right now. Just give me 1 second. Okay, screen share.
Thanks for your very generous introduction.
My pleasure.
It's true to say that I think the Nature journals have taken a big interest in synthetic biology. So very many great papers have been published at Nature, Nature Biotechnology, Nature Chemical Biology in particular, and of course, not to forget Nature Communications, where Ross Cloney, who leads a team there, has really championed this subject and is incredibly enthusiastic about everything synthetic biology. And this is just a really short presentation to just try and give you a little overview of how I think about synthetic biology. So I've been an editor for a long time now. I've been an editor for 20 years. And during that time, I've been at a reviews journal. I was at Nature Reviews Micro. And I've also been at Nature Biotechnology, which has published some of the very best synthetic biology papers, I think, especially because it's a technology journal. Of course, there are very, very many other journals that have specialized in and published some really fantastic work, including metabolic engineering and the ACS journals, synthetic biology, and other journals too. So if we could just go on to the next slide, please. I just put together a really quick visualization of how the field is growing, and this is just from doing a PubMed search for anything that has synthetic biology in the title or the abstract. And in 2007, there were very few of those papers, but last year there was just under 1,400. And of course, this is just a subset of what's out there because that's a very small search term. And I did my PhD in 2003, so I've watched this field grow from the outset. And I think one of the things that as editors we love about synthetic biology is that everyone is so innovative. They are so generous with their time in reviewing each other's papers. And there are lots of really great moves to make everything open in synthetic biology, including standards of reporting, including data sets, including code, and in sharing parts using services such as AdGene, for example. So I think it's a really collaborative exciting field, and it's always been obvious that there's going to be loads of amazing applications coming out of synthetic biology. And I think that's starting to come to fruition now. I've just picked out a few papers to kind of reinforce that point. So if we could go on to the next slide, please. So many years ago, Nature Biotechnology published a focus issue on synthetic biology. So this was in 2009. And in it, they had a really nice feature where they asked a few leaders in the field at that time what synthetic biology meant to them. And I think out of all those definitions, the one that George Church brought resonated the most with me, because there are so many different aspects of this field. There's genetic engineering, there's genome engineering, there's modeling the minimal unit of life, there's trying to replicate things, you know, like how a jellyfish swims or something like that. There's materials design. But synthetic biology is kind of the meta collection of all these really fascinating fields. And at its heart, it has to have interoperable parts which can be used to engineer all different types of life forms and processes. So can we go on to the next one, please? So this is the first really big news synthetic biology paper that I remember. And this was when they were able to synthesize artemisinin in yeast. And this is a big deal because it's a plant-derived product and it's very difficult to guarantee large and pure yields of this key antimalarial from plants. And so this was a really big breakthrough, and you can see just how many views that paper has had since it was published some time ago, and from the laboratory of Jay Kiesling and others. And if we go on to the next one, this was 10 years ago. This is their most recent paper coming out in Nature again, where they had solved the problem of how to produce this anti-cancer drug vinblastine by metabolic engineering. And again, this is another plant product, and plants are just such an amazing natural resource of molecules that can be used as therapeutics, but of course they're not always so easy to work with. And then you've got issues of land use being devoted for harvesting plants just to purify therapeutics. And if you can do it in microorganisms, you reduce the pressure on the land, and you also guarantee purity of the compound. So this was another really incredible multi-step process that was engineered to produce a key therapeutic. If we can go on to the next one. And it's not just metabolic engineering that's caught my eye. So there have been some really amazing computational advances. And I've picked out here Cello, which is a set of programming language specifically for designing computational circuits in living cells. And originally this was devised for E. coli, but they've expanded it to yeast and I believe other organisms too. So these kind of advances mean that it makes it easier for the synthetic biologists and engineers out there to design circuits that will work really well in cells. Can we have the next one now? And I think the most exciting paper I've seen in recent years is this one from Nature Biotech, where they managed to engineer organisms, Clostridium in this case, to grow using the excess waste gases from a steel mill and convert that into usable products at scale. They had a scaled up pilot plant here, and the whole process is carbon negative. So if we go to the next one, in a really nice news and views, the authors laid out how this promotes a circular carbon economy. And I think this really has been promised for so long in synthetic biology circles at conferences, but this is the kind of culmination of that. And I just thought this blew me away, because it's just absolutely amazing paper where they've gone from the engineering right to the production, and they've shown us what synthetic biologists can do to help with real life planetary problems. And if we can go to the next one. Is that the last one? I think it might be the last one, actually. So I think my point overall was just to try and convey a bit of my excitement about the field from all very many points of views, but particularly from my viewpoint in Nature Micro now, where we're really trying to publish microbiology that will help society tackle the Sustainable Development Goals. And so that was the note I wanted to end on.
Okay, so wonderful. Thank you. I glad because I see so many people, and actually almost all of them actually participate in this seminar series as a pioneer speaker, including Jay Kiesling, Chris Voigt, and also Ranjatek. I know the Mike very well because we are currently editing one journal for the special thing, and also absolutely brilliant work about carbon capture because that would address the problem of climate crisis, and we are very not good situation at this moment, but I also optimistic because technological innovation would address those problems, by synthetic biology or metabolic engineering or systems biology, whatever people wanted to call it. And also the paper you just show, especially the Nature paper by Jay Kiesling group, that is the 30-step pathway. They put it together in East. And I was completely blown away after I stayed up in the conference talking with people until 5 a.m. And then Jay Kiesling gave the keynote talk about that stuff for the first time. And then that made me awake completely because I never seen any pathway engineering that is the more than 30 step. I mean, actually 30 step, exactly, they have, and then making the very important anti-cancer drug just biologically. So I'm very optimistic, even though we have so many global problems, but that's another reason I'm doing this one, because our future is basically rely on the young speakers, young researcher like today's speakers. Okay, thank you so much. I mean, that is absolutely brilliant, amazing talk, and that is a very insightful history of synthetic biology. Thank you. Okay, let me now introduce the main speaker of today with a longer introduction. Dr. Jinjin Diao is a postdoctoral scholar of my lab in Washington University in St. Louis. Despite my chairing the seminar series, interestingly, he is the only rising star speaker from this university and one of the 2 rising stars who have been nominated from this university, probably due to the lack of interest in synthetic biology in Washington University. The other speaker, unfortunately, decide to join the industry, leading to dropping out from the seminar series. Jinjin is focusing on valorization of waste, including lignin and plastic waste, as well as carbon dioxide. Of course, one of the most important topics in this century. He received his PhD from Tianjin University in China, majoring in biochemical engineering. He has demonstrated how microalgae can be reprogrammed to produce value-added chemicals from carbon dioxide as the carbon source. This means that he truly cares about our environment by reducing CO2 and addressing the plastic, industrial, and agricultural waste. I am probably biased because he's my advisee, but I believe he's truly a great person from not only a scientific perspective, but also as a hardworking dad and husband. In fact, he is a father of a 2.5-year-old baby. Unfortunately, due to the horrible COVID situation and US-China tension, he could not visit China and see his baby for 2.5 years in person. I hope this terrible situation will end soon and we will live in a better world. Jinjin, thank you so much for your time and your commitment to this community to contribute to solving lots of problems, including climate crisis, and the podium is all yours. And thank you again.
Thank you. Thank you for your introduction. And okay, can I share my slides now?
Yes, yes, please do so. Wonderful.
Okay. Let's start. Thank you, Dr. Moon, for your very for your introduction of the situation I meet now, and it's I really missed my family very much, but I really want to like finish the fantastic project which is I'm working on as soon as possible, and I will show here today to you to demonstrate what I'm doing now. And okay, let's start. And first of all, I would sincerely thank our SynBYSS chair, Dr. Tae Seok Moon, who is also my postdoc advisor, organized this amazing community for young people. Also, I would like to thank my pioneer speaker, the chief editor of Nature Microbiology, Susan. I think your insights will inspire more young people to committed to the development of synthetic biology. And today I'm going to talk about my recent progress, which is about how to turn wastes into values through metabolic engineering and synthetic biology. Humans affect regional and environments through the agriculture, development, and industry. And in these activities, lots of wastes are generated that have been considered as threats to the safety of our ecosystem. For instance, the emission of greenhouse gases is the invisible hand of the global climate change. The widely use of non-degradable plastics cause wide pollution and the increased accumulation of toxic byproducts from the industry. And even more and more people choose low carbon lifestyle. A more efficient way to deal with the waste is still urgently needed. Unlike reduce, the reuse strategy could clean up the waste streams and then turn the carbons into other forms of chemicals, which can enable us to have a double win and make our planet more sustainable. Carbon dioxide is a primary greenhouse gas emitted from human activities. And in 2020, carbon dioxide accounted for 79% of all US greenhouse gas emissions from human activities. Thus, the development of new strategies to capture carbon dioxide is very important. Inspired by the nature photosynthesis, in 2009, James Liao published their work in Nature Biotech, which is talking about the development of cyanobacterium, Syneococcus elongatus, PCC 7942 as the chassis for upcycling carbon dioxide to the isopropanol. And then some other cyanobacteria have been adapted for the production of various forms of biofuels. And also our peer speaker, Bo Wang, who have already given us a beautiful story about how to develop a cyanobacteria chassis for upcycling, for converting carbon dioxide into degradable plastics. And these advanced studies hold great potential for humans to reduce the accumulation of carbon dioxide in the atmosphere. And even with advantages, the most challenging thing is that the production of chemicals from cyanobacteria chassis is usually low, and which make the overall process economically non-profitable. So instead of these fuels and bulk chemicals, we also want to produce some high-value chemicals from carbon dioxide. Here, I just want to talk a little bit about our efforts in producing high-value chemicals from carbon dioxide. Because cyanobacteria intracellularly accumulate large amount of photosynthetic pigments, which can be the precursors for the biosynthesis of astaxanthin. And we thus engineer the cyanobacteria to sequester carbon dioxide for the production of high-value chemical astaxanthin. Surprisingly, under regular growth condition, our engineered strain could accumulate up to 30 milligram per gram dry CO weight after 10 days of cultivation, which is similar to the native producer, Homitococcus. In addition to the carbon dioxide, the solid waste streams like industry biorefinery, bioproduct lignin, and the non-degradable plastic are also the problems we need to address. Because lignin and plastic are carbon-rich materials, we also believe that those plastics, those two forms of waste can be developed and used as feedstocks to fuel the microbial cell factories for the production of value-added chemicals. But the main challenge is how can we? Let's first talk about lignin. Lignin is the second most abundant polymer on Earth, which holds promise as a renewable feedstocks for the production of fuels and platform chemicals. However, as a byproduct of the biorefinery industry, lignin is usually treated as waste because the depolymerization of lignin typically results in diverse aromatic products, which are challenging to valorize. So far, with the implementation of microorganisms that have multiple types of funneling pathways, lignin can be converted into various chemicals such as biopolymers, biofuels, which paves a new way for lignin valorization. Among the microbials, Rhodococcus PD630 has been considered as a promising chassis for valorizing lignin because it has high aromatic tolerance and utilization efficiency. It can accumulate a high content of lipid, and its genome has been sequenced, and we have developed multiple genetic tools for this strain. Previously, by using adaptive laboratory evolution and transcriptomics analysis, we have identified the putative funneling pathways for the 5 model aromatic compounds, including benzoate, hydroxybenzoate, phenol, vanillin, and guaiacol. Notably, it is quite common that in the aromatic degrading microbials, the aromatic degradation pathway are always tightly regulated. Even though we have identified the funneling pathways in Opacus, how this strain regulates the individual funneling pathways and how the different funneling pathways interact with each other when additional aromatics available are still unclear, which restricts the further optimizations of this chassis for lignin valorization. From the previous whole genome sequencing results, we have found different families of transcription factors that are located adjacent to the proposed aromatic funneling pathways. However, the specific roles of those transcription factors are still unclear, and we thus try to analyze the functions of those transcription factors by using synthetic biology tools. It is very interesting to find that our strain has 2 phenol funneling pathways that are distributed in the different regions of its genome. Here we name them Path 1 and Path 2. For PAS1, we can find an ARAC type transcription factor is located adjacent to the two-component phenyl hydroxylase. To test if this ARAC type transcription factor was involved in regulating the phenyl degradation, we first knock out this transcription factor, and then we placed the upstream promoter region of this cluster in front of GLP, and the resulting construct was expressed in both wild type and the transcription factor knockout mutant. And our results showed that the promoter response to phenol was completely off, and the phenol consumption was also abolished. And this result showed that in regulation this phenol funneling pathway, this ARAC type transcription factor works as a transcriptional activator. For the PAS2, in the transcription factor knockout mutant, the promoter activity of the two-component phenyl hydroxylase was severely inhibited, which indicated that this ARAC type transcription factor may also work as a transcriptional activator. However, the phenol consumption profile of the mutant was almost identical to the wild type strain, suggesting that this phenol funneling pathway might be not essential in this strain. As those two transcription factors are pretty similar, we next conduct cross-validation to test whether those two transcription factors have crosstalk or not. The results showed that knocking out each of the two adjacent transcription factors had no effect on the induction of the non-concatenate cluster. Additionally, we also generated a dual knockout mutant via homologous recombination, and interestingly, the fluorescence output of the promoter PLPD6740 was null, suggesting that this promoter may have crosstalk with the transcription factor LPD6574. Considering these results, we hypothesized that PAS1 is the essential one for phenol consumption in Opicus. To test this hypothesis, we first generate another mutant by knocking out the structure gene, which is LPD674, which is the LPD6740 in the PAS2. As expected, when feed 0.7 gram per liter phenol as the soil carbon source, the wild type and the mutant showed statistically identical cell growth. However, even with repeated attempts, we cannot disrupt the structure gene LPD6575 in the PAS1. Thus, a T7RAP based CRISPR-I platform was applied, and the results showed that the downregulation of LPD6575 significantly inhibited the cell growth when using phenol as a soil carbon source. And these results suggest that PAS1 is the essential pathway for the phenol consumption in our strain. And similarly, by using this GLP reporting system, we have identified the transcription factors that are involved in the regulation of hydroxybenzoate, vanillin, and guayacols funneling pathway. And our results showed that all those transcription factors are transcriptional activators. Now, however, the transcription factor in regulating the benzoate funneling pathway is still not founded. And the funneling pathways we described above could convert the aromatic compounds into the intermediates like catechol and PCA for the subsequent degradation by the beta-keto adaptive pathway. In PD630, the fundamental roles of both catechol and PCA branch of the beta-keto adaptive pathway have been confirmed by gene knockout experiments. However, the transcriptional regulation of those two pathways is still unclear. So for the catechol branch, the critical genes in an ICLI type transcription factor are clustered, but are transcribed divergently. Previously, we have found that the promoter of the catechol dioxygenase could respond to phenol, benzoate, and guaiacol, suggesting that the real effective compound should be the intermediates of the catechol degradation pathway. To test this hypothesis, we first selected and tested catechol and mucinate in the wild type strain. Analysis showed that promoter of the catechol dioxygenase displayed only low basal activity in the glucose condition, but the promoter activity was markedly enhanced in the treatment of catechol and milknate. As it is known that catechol can be spontaneously converted into milknate by the basal expression of the catechol dioxygenase, thus suggesting that the real effective compound may be the milknate. To analyze the function of this ICOR type transcription factor, the GFP construct was expressed in both wild type and the corresponding knockout mutant. Our results showed that in the knockout mutant, the promoter activity of the catechol dioxygenase was strongly increased with cultivation on both glucose and the respective aromatic compounds, indicating that this ICOR type transcription factor works as a repressor. Based on the analysis of the sequence alignment, we found that the -35 and -10 region of the transcription factor's promoter are overlapped with the transcription factor with the putative transcription factor binding site, which suggests that this ICOR type transcription factor may also regulate its own expression. To test this hypothesis, the GFP construct was expressed in both wild type and the corresponding transcription factor knockout mutant. Our results showed that in the wild type strain, the addition of milknate significantly reduced the promoter activity of LPT6569. And in the knockout mutant, the output of the fluorescence was completely off. And these results indicated that the expression of this transcription factor is regulated by autoregulatory activation. And we thus figured out the regulation pattern of this cluster, that is in regulating the catechol branch of the beta-keto-adapted pathway. This transcription factor works as a repressor, and in regulating itself, this transcription factor works as an activator. In addition to the catechol branch of the beta-keto-adapted pathway, the PCA branch has been identified as a key pathway for the degradation of hydroxybenzoate and vanillin in Opacus. In contrast to the related Corynebacterium, only one ICOR type transcription factor, which is LPD5454, is located in this operon. To investigate how this single transcription factor regulates the entire PCA operon, the intergenic DNA fragment of the divergently oriented coding regions were cloned in front of GFP and in both orientations and expressed in both wild type and the corresponding transcription factor knockout mutant. Our results showed that this ICOR type transcription factor works as a repressor in regulating this entire PCA operon in our strain. Bacteria often degrade the mixed compounds sequentially. This type of ordered catabolism termed carbon catabolite repression. Previously, our study has revealed that this strain preferred benzoate and hydroxybenzoate over phenol, guaiacol, and vanillin. To investigate the adaptability of Opacus to the presence of multiple aromatics as carbon source, the effects of hydroxybenzoate on the three other model compounds catabolism were examined. Our time course analysis of the CO growth and the aromatic concentrations showed that the presence of hydroxybenzoate inhibited the catabolism of phenol and guaiacol, but not for the vanillin, because the funneling pathways play significant roles in the degradation of aromatics. When we look at the responses of the transcriptions of the phenol, guaiacol, and vanillin's funneling pathway, we can find that the presence of hydroxybenzoate significantly inhibited the induction of phenol and guaiacol's funneling pathway. However, in the vanillin scenario, hydroxybenzoate did not cause a significant delay in the vanillin induction, which is consistent with the consumption profile. We also try to analyze the hierarchical utilization between hydroxybenzoate and guaiacol as an example to investigate the potential molecular mechanism. In the wild-type strain, the induction of guanyaco's funneling pathway can be inhibited only when additional hydroxybenzoate was available. This result suggested that rather than the intermediates in the hydroxybenzoate degradation pathway, hydroxybenzoate itself participates the observed transcriptional repression. Transcription factor-based induction prevention later describes the preferential utilization of benzoate over hydroxybenzoate in other strains. To validate whether the preferential utilization of hydroxybenzoate over guaiacol is also driven by this mechanism, we generate two different mutants. And the first one is the transcription factor knockout mutant, which is delta LPD 6765. And the other one is the hydroxybenzoate monooxygenase knockout strain, which is delta LPD 6764. However, the repression was abolished in both mutants, suggesting that diverse carbon catabolite repression mechanisms have evolved in this bacteria to deal with the mixtures of several substrates. And of course, and this idea, of course, needs to be further investigated. And after we get a better understanding of the aromatic degradation pathways in Opacus, next we are trying to use this information to guide the lignin valorization. And milknate, which is the precursor for the synthesis of polymers such as nylon, is typically produced from petroleum. As milknate is the intermediate of the catechol degradation pathway, knocking out the milknate siloisomerase would enable Opacus to accumulate milknate. But in this case, if benzoate was the sole carbon source, CO will die, as no carbon can be filled the TCA cycle to support the CO growth. Instead, we choose hydroxybenzoate as the second carbon source to support the CO growth, as hydroxybenzoate can be degraded through the PCA branch of the beta-keto adapted pathway. And in this case, our results show that benzoate can be converted to the milknate at the carbon yield reaching up to 0.77. Another application I want to show here is to develop a kill switch system in Opacus based on the aromatic sensors we identified. As we know, many pollutants are phenolic compounds, and our strain is able to tolerate and degrade aromatics, which has the potential to be developed into artificial bactericide. But the uncertainty of the genetic engineering strains in the open environment is still a big concern. To address this, we want our bacteria controllable. Which means after the degradation task complete, cells will suicide. To achieve this goal, we develop a kill switch system in Opacus. We used the aromatic responsive promoter was used to drive the expression of the TAT regulator, and the TAT promoter was used to drive the expression of the toxin protein. And when the aromatic compounds on, the TAT regulator expressed and repressed the expression of the toxin protein. And on the contrary, once the aromatic compounds deplete, TAT-R's expression stopped and the expression of the toxin protein becomes constitutive and the cell dies. It is very interesting to see that after we systematically tune this device, we can achieve 100% killing efficiency in Opacus. Next, I'm going to talk a little bit about our opinion regarding the PET recycling. Because of the versatile applications such as single-use packaging, clothing, PET is being of the most abundant polyester plastic manufactured in the world, with a global production exceeds 17 million tons. But only a minor fraction of that volume is recycled, which presents as one of the most challenging ecological issues for us now. It is quite a common sense that we can make PET from waste PET. Specifically, the waste PET can be depolymerized into monomers, and the purified monomers can be repolymerized into new PET. This strategy can be called PET to PET recycling. However, the biggest problem of this recycling strategy is that the process is cost-intensive when compared to the virgin PET, making it economically somehow non-profitable. Inspired by the lignin valorization, we also believe that if the monomers from the depolymerized PET can be used as feedstocks for microbials to produce value-added chemicals, PET recycling would be made more economically attractive. But the challenging is to find a robust microbial chassis which has the ability to use PET breakdown products as the soil carbon source. To this end, we screened several representative soil bacteria for growth on both monomers TPA and AG, because soil bacteria are promising decomposers for many types of hydrocarbons. Luckily, one rhodococcus species, here we named RPAT, could consume both type of monomers. To test the feasibility of using RPAT as a microbial chassis for the upcycling of PET, we prepared equimolar mixtures of TPA disodium salt and EG, ranging from 40 millimolar to 300 millimolar each to mimic the PET hydrolysate. When the concentrations of these simulated hydrolysates increased from 40 to 100 millimolar each, RPAT showed an increased final cell density with no significant difference observed in the durations of the lag phase. In contrast, when exposed to higher simulated hydrolysate concentrations, cell growth was obviously inhibited, suggesting that the bacteria could readily tolerate no more than 200 millimolar sodium. Next, we tested the cell growth by using PET alkaline hydrolysate as the soil carbon source. We found that the cell growth of RPAT was uninhibited when exposed to tenfold diluted PET hydrolysate, where the sodium concentration was about 200 millimolar. And these results indicated that RPAT would be an ideal biological chassis to pair with PET alkaline hydrolysis for the biotechnological upcycling of PET. With this promising chassis in hand, next we are trying to engineer it to produce some chemicals from waste PET. When we look at the genome of RPAT, we found it has a complete carotenoid biosynthetic pathway, and a tiny amount of lycopene was detected in the wild-type strain. And 3 unknown carotenoid compounds were detected as well in HPLC analysis, which is interesting here. In order to improve the lycopene production, we first tested and established the arabinose-inducible overexpression system in RPAT. And then the PPAT promoter was used to drive the expression of the CRT-EBI operon, which contains the 3 critical genes of the lycopene biosynthetic pathway. And we found that in strain S1, with the addition of arabinose, the production of lycopene was increased to 600 microgram per liter. And from the HPLC analysis, we also found that the peak area of the 3 unknown carotenoids was significantly decreased in the strain S1, suggesting that the biosynthesis of these 3 unknown compounds competed with the synthesis of lycopene. Interestingly, a closer examination of the native carotenoid biosynthetic pathway revealed the putative lycopene beta cyclase may catalyze the competition biosynthetic pathway. Consequently, we replaced the CRT-LB gene in strain S1 with a hygromycin-resistant selective marker, which generated strain S2. Although carotenoid analysis revealed that the 3 unknown compounds had been eliminated in strain S2, no changes in the lycopene production was observed, no matter we add arabinose or not, indicating that this level of lycopene production was independent of the synthetic CRT-EBI operon. To validate this hypothesis, we knock out the CRT-LB gene in the wild-type strain, which resulted in strain S3, and we found about 500 microgram per liter lycopene was produced in this mutant, which supported our hypothesis that the deletion of the CRT-LB LB gene contributed to the lycopene accumulation in strain S2. And these results also indicated that in strain S2, the key factor in determining its lycopene production likely was the supply of the precursors. To enhance the carbon flux of the native MEP pathway, two key genes were selected and overexpressed in strain S3. And we found that the co-expression of DXS and IDI genes significantly increased the lycopene production. However, interestingly, when the DXS IDI and the CRT EBI operons were co-expressed in strain S7, we found the lycopene production was decreased, which might be attributed to the metabolic burden caused from these two plasmid overexpression system. At last, to cement the proof of concept of this upcycling of PET to value-added chemicals, we used the real PET hydrolyzed and strain S6 was inoculated into minimal medium with 10% crude PET hydrolyzed as a soil carbon source. The final lycopene titer was approximately 1,200 microgram per liter, which was more than 500-fold higher than that in the wild-type strain. With this achievement, we believe that biological conversion holds a great potential as a means of achieving post-consumer PET upcycling. And at last, in summary, and like we have mentioned at the beginning of this talk, wastes have tremendous amounts of carbons, and we believe that with the development of synthetic biology, more robust chassis will be developed to convert the wastes into other forms of chemicals, like bulk chemicals, biofuels, pharmaceuticals, which will play a critical role in achieving President Biden's goal of net zero emissions by the middle of this century. And we also believe that this will help us to save our planet a little bit more. And at last, I would like to thank all the members from Dr. Moon's lab for supporting my research. And I would also like to thank the funding from Department of Energy. And at last, I'm happy to take any questions if you have. And thanks again for your time and attention. And thank you, everyone.
Thank you so much for the wonderful talk. I already have a question from Sujan. If you wish, you could speak and then ask question. Challenging one, please.
Well, it may be a very naive question. It was a really well put together presentation. Thanks for spending the time to do that. I was wondering whether Rhodococcus is used in any industrial processes already, or are you working on it in Rhodococcus and then thinking of moving the whole thing into a more industrially relevant strain?
I think some group is trying to pursue the Rhodococcus to be applied in the industry, because I know some group try to use Rhodococcus species to produce milknate from lignin to produce the precursors for the plastic synthesis. So I know this species, of course, holds great potential to be developed into a strain which can be used in the industry application. Yeah.
I suppose the issue for industry is that they want to have strains that they have already optimized the growth conditions of. I'm imagining that's the case. I don't really know for sure. So I think if you can show that your pathway will also work in an industrial microbe and that you can control it and optimize that too, then it will show that it's really got the potential for translation.
Yeah, I agree.
Yeah. Obviously, I don't know how easy that is. Maybe it's really difficult.
Yeah, sometimes it's difficult.
Yeah. So are there other questions?
So do you have another question, or that's your question? That's all of your question.
I could ask more questions. I just wondered if anyone else had questions. I don't want to monopolize.
Sure, you could ask the question, but to answer your question only because I'm his advisor, it's a kind of one of benefit having advisor as host of the seminar series. So not exactly same species, but the genus of Rhodococcus, some of that actually used in commercial production of some chemical, if I remember correctly. And important thing is the strain he used, all of that is basically the BL, safety level 1 strain. So very safe, something like the E. coli, the lab strain. But this is originally isolated from soil environment contaminated with the toxic chemical. So that is naturally has the huge, a lot of ability in terms of tolerating toxic chemical. That's why this is a brilliant compound. I mean, the organism. At the same time, this one accumulate lots of lipid up to 80%. That means the future potential biodiesel producers. So that's why I love this bacteria a lot.
Yeah.
You could ask another question because I don't see any other question from the chat or Q&A.
I'm wondering about the lignin, I suppose. Have you are you able to show that your organism can degrade lignin at various stages? What sort of pre-treatment's needed?
Yeah, we before we use the lignin, we need to pretreat the lignin to depolymerize the lignin into the monomers. But our strain, I don't think the strain we use can naturally depolymerize the lignin. So if we want to use this strain, we need to have some pretreatment process to break down the lignin products first.
So I think that's one of the issues, isn't it? That it would be ideal if organisms could use feedstocks that were required fairly little pre-treatment, because then the overall cost would be lower and the barrier to using the organism would be lower too. But I don't really know how do you know much about how it works in soil? Do you know that this operon is switched on in soil, for example, or have you done all the work in the lab?
I know there is a group in US who try to overexpress the engineer our strain to secrete an enzyme which can be used to depolymerize the lignin then the produced monomers can be directly used by the bacteria. And I guess this kind of process will decrease the overall costs when we use these bacteria to do the lignin valorization. But I think this we are also trying to engineering our strain to squeeze some enzymes we are interested in, and we also think this kind of process will make our bacteria more attractive to other researchers. Yeah.
You mean if it can synthesize a variety of products, not just ethanol? Yeah.
Yes.
So where in the soil where you isolated this from, is it likely to be growing in a community that maybe includes some fungi? I'm just wondering what in the natural environment might break down that lignin for the Rhodococcus to use.
Yeah, if we can, like in the natural environment, the bacteria forms like a consortium, and the fungi put some proteins to help to depolymerize the lignin, and the bacteria may consume the monomers to support the cell growth. And inspired by this aspect, we are also in our lab, we are also have a huge interest to construct the microbial consortium to valorize the lignin and to upcycle the plastic, because we know the plastic is sometimes always sometimes a mixture. Like we have we have PET, we have PE, we have PP. That is a mixture of plastic. So maybe one bacteria is not able to degrade all the plastics. So we are now having a huge interest in like construct a microbial consortium to like valorize the PET or valorize the plastic or valorize the lignin. I guess this is very interesting because the consortium, the concept of the construction of microbial consortium is of great interest to the researchers in our field. And
yeah, I
really
completely agree that the whole field of synthetic ecology and trying to mimic community functions is really, really interesting. Although the only drawback seems that, from what I understand, industry prefers to have single organisms. They don't, not really as interested in communities, and I'm presuming that's because of issues of propagation and growth and maybe even safety. As you say, if you've got one that's generally regarded as safe, that's great, but if you've got a consortium with some unknown components in it, then that would be more difficult for industry. But it does seem that that would actually be really exciting if you could synthetically assemble a consortium that could then break down wood waste or plastic waste. That would be a very nice study. So that sounds
Yes.
really, really interesting. I don't think I've got any more questions now.
Yeah, you know what? Thank you so much for this insightful question. I mean, that's very exciting because you exactly kind of point out my future direction, that is consortium engineering. So we have the multiple paper under review. One of them is just talking about new tool development that enable microbiota engineering, very specifically strain levels. You could kill only one strain, or you could isolate only one. And also, interesting thing is, industry thinking about one single bacterium for the production, but to solve this global climate problem, I strongly believe we need to have complete paradigm change. And I actually had one article in your competitor journal, that is a trend in biotechnology, in the Cell journals. I had very radical idea. I call it soil as a huge bioreactor. That means the soil as not just thinking about just bioreactor, it's just small, whatever, 100,000 liter reactor, but I'm thinking about entire soil on Earth as the huge bioreactor that capture CO2 or nitrogen and then capture the carbon in the soil, and then potentially, we produce some chemical or material by converting those things into, some value-added one. And at the same time, thinking about adding so-called probiotic. The probiotic here means for the, Earth's health. So the probiotic could be fix nitrogen better or fix carbon better. In that way, reduce carbon better. And then at the same time, degrading those toxic chemicals, including, aromatic compound, also the waste. I'm talking about plastic waste. So why not? That requires, of course, biocontainment and also huge engineering effort, because the consortium engineering is not easy, but we are going, one step further toward that, goal, achieving that goal at this moment. So you will see lots of exciting work from my lab. And if you are interested, I can forward that paper to you. That is my provocative kind of idea, kind of presenting in one paper.
Yeah, I'd definitely be interested to read that. Is it a viewpoint or an opinion piece?
Yeah. All right.
Please do send it to me. I will read it.
Sure, I will do so. Okay, let me check one more time whether do we have any question not here, not in the chat. Okay, let me close. Okay, thank all for joining and staying today. We'll meet again next week on October 20, Thursday, the same time, the same Zoom link. We'll have Dr. Clem Fortman from Biomate, and that is one of the big organization supporting commercialization of synthetic biology research and DoD funded, and Dr. Caroline Mills from Northwestern University. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers, including the chief editor of Nature Microbiology. I'll promote you to panelists who can speak and show your handsome and pretty face. So thank again, and I will stop recording. Just give me 1 second. Stop recording.